all photosynthetic reactions, right from incidence of light on green parts of the plant to the synthesis of glucose. They are broadly divided into two phases. The light phase reactions, the dark phase reactions. In the last 2-3 lectures, we have discussed about the pigments, chlorophyll molecules, the light requirement, the light phase reactions, cyclic and non-cyclic photophosphorylation. Now, in this class, let us try to understand the second phase of reactions in the photosynthesis. They are often referred to as the dark phase reactions. In 1905, F.F. Blackman showed that photosynthesis consists of two types of reactions. The one is a rapid light dependent photochemical process, that is light phase reactions. A slower temperature dependent biochemical reactions, they are known as dark phase reactions. These are termed as light reactions and dark reactions respectively. Now, this diagram is very useful for overall understanding of the photosynthesis. The reactions take place in two phases, light phase reactions, dark phase reactions. If you take the light phase reactions, green leaves, mesophyll cells, chloroplast, inside the chloroplast, grana. Then light falls on the grana. Other requirement is water. And during the light phase reactions, oxygen is released as a result of photolysis of water, then protons, electrons. During this light phase reactions, ATP, energy rich molecules, NADPH, proton and electron carriers are synthesized. This is energy rich molecule, this is reducing power, together known as assimilatory power. Now, these are the products of the light phase reactions. These are required for the dark phase reactions in order to reduce the carbon dioxide to a carbohydrate. These reactions take place in the stroma of the chloroplast. You see here, carbon dioxide, which is in the oxidized state, which is inorganic, which is energy less or energy poor, is converted to a carbohydrate that is organic, that is energy rich, that is reduced compound. Oxidized to reduced, inorganic to organic, energy less to energy rich. Then where do all these come? They come from ATP. Required energy comes from the ATP. The reducing power is given by the NADPH. Therefore, energy rich carbohydrates are synthesized, glucose and starch. Now, another important point you have to keep in mind is, for photosynthesis, carbon dioxide is required. But carbon dioxide is absorbed only during the dark phase reactions, not in the light phase reaction. For light phase reactions, carbon dioxide is not necessary. Only the requirements for light phase reactions, light, water, chlorophyll molecule. But here, the carbon dioxide is required. So whatever the carbon dioxide that is taken in during the photosynthesis is taken during the dark phase reactions. So this is then after utilization the ATP is converted to ADP, NADPH is oxidized to NADP. Then light reactions, dark reactions, if you see the difference, main differences, these are light dependent, these are light independent, no light required, they are not dependent on light. Splitting of water molecule releases oxygen as a byproduct. Carbon dioxide is reduced to carbohydrate. Carbon dioxide, which is in the oxidized state, is reduced to a carbohydrate. Reactions take place in the grana of the chloroplast, these in the stroma of the chloroplast. The products of light phase reactions are ATP, NADPH2. Here, the products of the dark phase reactions, carbohydrates, water, oxygen. Then it takes place in the presence of light. Dark phase reactions, when we say dark phase reactions, it gives the impression that these reactions take place in the absence of light or darkness, but it doesn't mean like that. They take place during light, in presence of light and absence of light. In order to differentiate these reactions from this, they have called them as dark phase, but it doesn't mean that dark phase reactions take place in the darkness. Photochemical phase, biochemical phase, grana, stroma. Here it is NADP to NADPH. Then here this, the end products ATP and NADPH. Glucose is the end product. Then photolysis of water. All these are the main differences. Dark phase reactions, these are also called carbon assimilation pathway. Carbon, which is present in the carbon dioxide, which in turn present in the atmosphere, is assimilated into the plant. Carbon dioxide is present in the atmosphere, it is absorbed by the plant, it is in organic form, it is assimilated into the form so that it is utilized by the plant and later on by different living organisms. Three types of carbon assimilation pathways have been identified. The dark phase reactions or carbon assimilation pathways in different plants take place in slightly different way. In some plants, C3 pathway, in some plants, C4 pathway, in some plants it is called crassulacean acid pathway or CAM, C-A-M, CAM pathway, CAM metabolism. CAM means crassulacean acid metabolism. C3, C4, then crassulacean acid metabolism. This is also known as dark carbon assimilation. Now let us try to understand C3 pathway. This is also known as Calvin cycle. C3 pathway is also known as Calvin cycle. Calvin, the scientist name. The reactions was worked out by Melvin Calvin, an American biochemist, along with his students Benson and Basham, for which Calvin was awarded the Nobel Prize in 1961. On his name, it is known as Calvin cycle. C3 pathway is also known as Calvin. The reactions take place in cyclic manner. Calvin, scientist names. Why cycle? The reactions take place in cycle. Whatever the compound that first participate is regenerated. So all the reactions ultimately take place in the cyclic manner. The reactions were worked out while working with chlorella. Calvin, while working chlorella, unicellular alga. Chlorella, unicellular eukaryotic alga. Then they have used, Calvin and his associates used C14 carbon dioxide isotope. Normally C12, but they have used C14. Technique was autoradiography. The technique used by them, autoradiography. The reactions are basically reductive type. Reduction of carbon dioxide to carbohydrates, organics. Calvin cycle is also known by reductive pentose pathway. Reductive pentose pathway. There are many pentose sugars in the Calvin cycle. Calvin benson Basham pathway, CBB. Remember C3 pathway, Calvin cycle, reductive pentose pathway, CBB cycle. In this cycle, first CO2 is accepted by ribulose 1,5 biphosphate, RuBP, commonly known as. The carbon dioxide acceptor is RuBP, ribulose biphosphate, a C5 compound. It consists of 5 carbons. First stable compound is phosphoglyceric acid. Phosphoglyceric, PGA. Then enzyme catalyzing the carbon dioxide fixation is RuBP carboxylase. RuBP carboxylase. It is also known by RuBP carboxylase or oxygenase. It is also known as RuBiCO. The enzyme that catalyzes the fixation of a carbon dioxide along with RuBP is RuBiCO or RuBP carboxylase or RuBP carboxylase oxygenase. This is the most abundant enzyme in nature because all are green plants, all perform photosynthesis, all of them fix the carbon dioxide. The most abundant enzyme in nature is RuBiCO. The first reaction is carbon dioxide fixation, that is called carboxylation. Fixation of the carbon dioxide is carboxylation. Calvin cycle uses the reducing power produced. What is the reducing power? ATP and NADPH from the light dependent reactions. Calvin cycle or C3 cycle requires the products of the light phase reactions ATP and NADPH. The cycle has 4 main steps: carbon fixation, reduction phase. First phase is carbon fixation, second is reduction phase, third carbohydrate formation, fourth regeneration. Carbon fixation, reduction phase, carbohydrate formation, regeneration phase. Then some of the reactions in the Calvin cycle: 3 carbon dioxide, 6 NADPH plus this is NADH reduced, 9 ATP, giving rise to glyceraldehyde 3-phosphate, 6 NADP, 9 ADP, 3 water, 8 phosphates. This is the overall reaction of the Calvin cycle. 3 carbon dioxide, 6 NADPH2, 9 ATP, giving rise to glyceraldehyde 3-phosphate. It is 3 carbon atom compound. The first stable compound is 3 carbon atom compound, therefore it is known as C3 cycle. 6 NADP, 9 ADP and 8 phosphates. 1 phosphate is given to this. Carbohydrate products of the Calvin cycle are 3 carbon sugar phosphate molecules, triose. In Calvin cycle, what is the first product? Strictly speaking, it is not glucose, it is not starch, it is, remember, glyceraldehyde 3-phosphate, G3P, glyceraldehyde 3-phosphate. Many people think that what is the product of the glucose, but strictly speaking, the carbohydrate product of the Calvin cycle is glyceraldehyde 3-phosphate. Hexose, 6 carbon sugars are not product of the Calvin cycle. Later on, the glyceraldehyde 3-phosphate is converted to glucose, but immediate product is only glyceraldehyde 3-phosphate. Although many texts, many books list the product of photosynthesis as glucose. This is mainly a convenience to counter the equation of respiration where the 6 carbons are oxidized in mitochondria. Then carbon fixation. A CO2 molecule of atmosphere combines with 5 carbon acceptor molecule. Remember, acceptor is RuBP, ribulose 1,5-biphosphate. It is C5 compound, contains 2 phosphates at first position, at the 5th position, RuBP, acceptor. This step makes an unstable 6 carbonate compound, C5 plus C1 of carbon dioxide, C6. It makes 6 carbonic compound that is unstable. It is formed and immediately splits into 2 molecules of 3 carbon atom compounds, 3-phosphoglyceric acid, 3-PGA plus acid. This reaction is catalyzed by the enzyme RuBP carboxylase oxygenase or RuBiCO. So, here the formula is CO2 plus RuBP giving rise to 2 molecules of phosphoglyceric acid. 1 carbon from carbon dioxide, 5 carbons from RuBP make 2 molecules. Phosphoglyceric acid contains 3 carbons. 2 molecules of phosphoglyceric acid. Reduction phase. In the second step, 3-phosphoglyceric acid is reduced to 3-phosphoglyceraldehyde. Acid, aldehyde. Acid is converted to aldehyde. It is in oxidized state, it is in reduced state. So, for this purpose, ATP and NADPH are required, which are the products of the light phase reaction. This stage gets its name because NADPH donates electron or reduces the 3 carbon intermediate to 3G3P glyceraldehyde 3-phosphate. Reduction stage steps involved in the utilization of 2 molecules of ATP for phosphorylation, 2 molecules of NADPH for reduction of every CO2. If one CO2 has to be fixed, this is the requirement, 2 molecules of ATP and 2 molecules of NADPH2. Regeneration phase. Some glyceraldehyde 3-phosphate molecules go to make glucose. We require the glucose, and while others must be recycled to regenerate RuBP acceptor. So therefore it is called regeneration. Regeneration phase requires ATP for phosphorylation to form ATP and involved in complex network of reactions. So thus, for every CO2 molecule entering the Calvin cycle, 3 molecules of ATP, 2 molecules of NADPH are required for every 1 carbon molecule to be fixed. To make 1 molecule of hexose, 6 turns of the Calvin cycle are required. The Calvin cycle has to take place 6 times in order to give out 1 glucose molecule as a net product. For every 6 cycles, 1 glucose, remember. Hence, for every 1 glucose molecule, 18 ATP, 12 NADH are consumed. This is also important. Then, overall, if you see, in Kelvin cycle, input 6 CO2 from atmosphere, 18 ATP, 12 NADP. Then what comes out of the Kelvin cycle? 1 glucose molecule, 18 ADP, 12 NADP. Because these are oxidized to this, then this ATP undergoes the hydrolysis to produce the ADP and phosphate. Input, output of the Kelvin cycle. So this is simplified Kelvin cycle. Ribulose 1,5 biphosphate, very commonly known as RuBP, C5 compound. This accepts the carbon dioxide and water. This phase is known as carboxylation. The reaction is catalyzed by RuBP carboxylase or RuBiCO. Then 3-phosphoglycerate, first stable compound. Then it is reduced to 3-phosphoglyceraldehyde, and from that, so for this reduction process, ATP and NADPH are utilized. They come from light phase reactions. Glyceraldehyde 3-phosphate. So this is actually the immediate product of the Calvin cycle. Later on, the glyceraldehyde is converted to sucrose, starch, glucose, like that. Only 1/6 of the glyceraldehyde 3-phosphate is utilized for sucrose. The remaining 5 are utilized for the regeneration of ribulose 1,5 biphosphate. This is called regeneration. For this, ATPs are required. So only 1/6 of the glyceraldehyde 3-phosphate is utilized for glucose synthesis. Therefore, in order to synthesize 1 complete glucose molecule, this entire Calvin cycle has to take place 6 times, 6 cycles. So thus, carboxylation phase, reduction phase, regeneration phase. Fixation of carbon dioxide, reduction of phosphoglycerate to glyceraldehyde, then production of the glucose, then regeneration. So this is again an elaborate Calvin cycle. We start from here. RuBiCO, 1,3-phosphoglycerate, glyceraldehyde, DHAP dihydroxyacetone phosphate. So glucose. So through various intermediates, pseudoheptulose, erythrose phosphate, pseudoheptulose 7-phosphate, ribose, then ribulose, xylose, RuBP is regenerated. This is a regeneration phase. This is carboxylation phase. This is a reduction phase. Glyceraldehyde, this is actually reduction phase. This is the production of glucose. Detailed Calvin cycle or photosynthetic. This is about dark phase reactions, Calvin cycle, C3 cycle, CBB cycle, reductive pentose phosphate pathway, wherein carbon dioxide acceptor is RuBP, enzyme RuBiCO or RuBP carboxylase oxygenase, because it can accept oxygen also, therefore it is oxygenase. Not only carbon dioxide, it accepts oxygen also. In some cases, later on we understand that point. Then reduction phase, the regeneration phase. Thank you. So in the next lecture, we will study about the dark phase reactions C4 pathway. That is also very important. In some plants, not C3 but C4 or sometimes both.